Morning everybody. Today we are discussing the next topic, that is the definition of the asymmetric synthesis. In the last class we have discussed the stereospecific reaction, stereoselective reaction, and what is the role of the asymmetric synthesis in day-to-day life. So actually the asymmetric synthesis is a chemical process that directly produces an optically active compound from symmetrically constituted molecule without requiring the resolution of a racemic mixture. Resolution means it is a separative method and it is used for the separation of the DL pair. Consider an example, acetophenone. We know that the acetophenone is a optically inactive compound and there is no chiral center is present in the acetophenone. So if we are doing the ordinary reduction of the acetophenone by using sodium borohydride, we will get the two product A and B. So the difference between product A and B is that in product A, OH group is towards the observer, and in the product B, the OH is away from the observer. That is the only the difference, but the chemical formula of the compound is the same. So second example is the same acetophenone. If you are treating the acetophenone by using a chiral hydrating agent, we will get only one single product, that is the S-alcohol. So this is the difference between the ordinary chemical reaction and an asymmetric synthesis. So the reaction which is represented in the bottom of the slide is an asymmetric synthesis, whereas the top one is the ordinary chemical reaction. So the definition of the asymmetric synthesis is very important. Next we are moving into chiral pool. Next topic, that is the chiral pool. So chiral pool means it is very difficult to synthesize an optically pure compound from an optically inactive compound. So naturally we are synthesizing an optically active compound from a naturally available optically pure compound. So the chiral pool is a collection of a natural or enantiomerically pure compound, and the examples are the naturally available amino acids, and we know that there is only one variety of the amino acid is occurs naturally, and we can use the derivative of the amino acids and carbohydrate. So the chiral pool consisting of an optically active natural enantiomerically pure compound, and which include the amino acid, derivative of the amino acids and carbohydrate. Now the chiral pool synthesis is a very simplest and oldest approach for enantioselectively synthesis, and we know that there are only a limited number of the samples collection is there in the chiral pool. That's why it is an oldest method, and it uses an enantiomerically pure natural products as the starting material. So definitely our product is also pure compound and optically active. So chiral pool consisting of the ready-made chiral centers, and it must be pure natural product, usually amino acids, which pieces containing the required chiral centers can be taken and incorporated into the product. So for example, the aspartamate, which is prepared from S-phenylalanine and aspartic acid. See here, this is the example in which the first one, the product which is shown in the left hand side is the targeted compound. If we are doing the targeted compound by retrosynthetic analysis, this arrow implies the retrosynthetic analysis. Retrosynthetic analysis means it is a reverse process of the actual chemical reaction. So we are we are cutting the targeted molecule and we will get the starting material. So the retrosynthetic analysis we will discuss a little bit later. So at present we will doing the retrosynthetic analysis in the targeted compound and we will get the two product, and these two product are similar to the natural product or amino acid which is available in the chiral pool. Now we are moving into the one of the example, the S-ipsenol, which can be prepared from the S-leucine. You know that the S-leucine is an amino acid and this is the structure of the ipsenol, and here we can see that ipsenol is the targeted molecule, and if you are doing the retrosynthetic analysis we will get the derivative of the S-leucine and a Grignard reagent. So the derivative of the S-leucine can be prepared from the amino acid leucine, and if you are treating the derivative of the S-leucine, that is the hydroxyl group, hydroxyl acid, and it is treated with an THP, that is THP is used for protecting the hydroxyl group. Protection means actually if any starting material which containing more than one functional group, so if you are adding any reagent, the reaction will occur on the both the functional group. So in order to avoid, we have to protect the most reactive functional group and do the chemical reaction on the other functional group. So here the OH group is most reactive than that of the carboxylic acid, so it is protected by using the THP, and the reaction is occurs in the acid group, that is COH, by using alcohol. So naturally acid will converted into the ester, and after the esterification it is reduced by using lithium aluminum hydride and we will get a corresponding alcohol, and after that the next hydroxyl group is also protected by using the tosyl chloride, and now the both the hydroxyl group are in the protected state. After that we will treated with the acid and base treatment, and so we will get an epoxide, and the epoxide is treated with a required epoxide is treated with a Grignard reagent, so we will get the ipsenol. This is the total synthesis. This is the synthesis method for the ipsenol, and we will get only S-ipsenol, and we are preparing the S-ipsenol from a naturally available amino acid, that is S-leucine, and S-leucine are belong to the chiral pool. So I hope that the concept of the chiral pool and the definition of the asymmetric synthesis. Thank you.